All right, I think we are good to go whenever you are ready.
Okay, ladies and gentlemen, thank you for coming to listen to this symposium on computational biology. So since the establishment of CMSA about 8 years ago, we always like to cooperate with our Evergrande Center in Medical School and Brigham's Women's Hospital, head by Vijay and Arlene. And I'm very happy that we have this going on, this symposium, except that because of the virus, we have been delaying it. And finally, we have today's symposium. So I'm very glad that Vijay and our colleagues in Evergrande Center are helping, and Martin Herbert is heading it. So let me turn the turn the symposium to Vijay. Thank you. Hello, Vijay?
Yes, I'm here. Can you hear me?
Yes.
Yeah, I want to welcome you all and thank you for participation, and especially the speakers. I apologize, we had to delay it a few times simply because of the COVID virus. And we have a really good lineup of speakers. And I'm going to introduce Martin Hemberg, who is a junior faculty who has just joined us from England, and he will be taking care of most of the day's proceedings. Martin, can you please take over the proceedings? And
thank you very much, Vijay. Yes, again, thanks everyone for joining. Exciting to see so many people joining in on a nice day in early May to enjoy some computational biology. So we have two sessions today, which are interrupted by a brief lunch break. Over in the UK, they have a early May bank holiday today, but John Marioni has been kind enough to step away from his vacation duties and chair this first session on single-cell computational methods. And then after lunch, I will be chairing the second session on more general computational biology. So over to you, John.
Thank you very much, Martin. So I think it's optimistic to think that it would be sunny. You said it's a pleasant May day. Here the temperature seems to be about 8 degrees Celsius. What's that, like 55 Fahrenheit? And yesterday it started some hailstone, so it's lovely, typical summer weather in the UK. Anyway, it is indeed my pleasure to chair this first session of the meeting. And we have a series of, I think, really stimulating and interesting speakers lined up. And the first is a colleague and friend, Peter Kharchenko, local to Harvard. And he's going to be talking about some recent work. He's done some really beautiful work on developing computational strategies to look at spatially resolved transcriptomics data. So without any further ado, Peter, over to you.
Thank you, John. Just give me a second while I share screen and go through all the usual trouble. Do you see my slides all right?
You see the presenter view, so you need to flip it.
The presenter view. Okay, how about now?
All good.
Excellent. Okay, well, thank you for this opportunity. So as I'm sure you all appreciate, in computational biology, we have challenges grand and small, and this one seems like a small one on the scale of things, but it's, you know, it's taking us a while to get a handle on it, and maybe some of the lessons learned will be of interest to others, and plus I think it might be a useful tool. So, you know, we're used to sort of working in this kind of expression space and looking at t-SNEs and other UMaps, other embeddings, and every point is a cell. Well, here we also have these velocity movements, but we think about these transcriptional states in this sort of abstract kind of large dimensional space, and we project it on 2 dimensions just so we could try to make sense of it and see major features. And it's very exciting. It has been over, I think, past 5 years or so. But the thing that, of course, should be in the back of our minds is that these cells, they don't exist in vacuum. We pulled them out, we're dissociated the tissue usually, and we measured their states, but in reality, they came from a particular context, from a particular structure in the tissue. And what we're seeing is sort of an aggregate, perhaps signature of their own identity, plus the influence they've experienced in the tissue. And of course, those are of great interest in many biological problems and would like to be able to measure it. So I'm showing here a slide from one of our work where we used in situ sequencing technology from Mats Nils's group to basically slice the embryo and look at transcriptional states within the slice. This is for particularly for neurocrest tissue. Neurocrest cells, they delaminate during development and migrate through the organism. And so as they migrate through the body, they experience all sorts of signals, some of which presumably cause them to actually differentiate into different cell types. So very important, kind of critical for, let's say, studies of cell fate decision and development. And yet, without sort of high resolution picture, spatial picture of where these cells were, and presumably, what sort of signals they've experienced, we can't really understand why they end up going one way or the other. So there's a number of technologies being developed, and I'm sure you've seen a lot of these exciting papers, and I'll just mention some of them. So some of them kind of based on in situ sequencing, some of them on fluorescent hybridization, sort of highly multiplex single molecule fish, MERFISH, SIGFISH would be some of them. Other techniques take a different approach, sequencing based, such as SlideSeq from Evan McCosker's group. They all basically aim to capture this context. And so we've focused on different techniques for different projects, but today I'll mostly talk about what I call imaging-based spatially resolved methods, which are either in situ sequencing or fluorescent hybridization-based, fish-based. So they bring their own computational challenges. The advantages are always, right, we get the spatial context. The other huge advantage that I can't wait to have in my data is that we don't have to dissociate the tissue. Most of the tissue, unless you work with blood, you have to take it through these rather complex and often very severe protocols to dissociate the cells that changes the composition of the tissue, that also affects the state of the cells we're trying to understand. So these methods, you're able to either use fixed or frozen tissue, which is a big advantage. But the challenges are also obvious. There are experimental challenges. These are kind of groundbreaking, tough techniques, can be tough to set up. A lot of these protocols are targeted, so we have to sort of pick a panel of transcripts that you will be analyzing, and the size of that panel can also vary. There's typically a trade-off between sort of sensitivity and the specificity of these assays. The particular problem that we focused on in our measurements is that the cell boundaries are not obvious, right? So we get very high resolution, we can see sort of nuclei structures, but the extent of the cell and the outer sort of membrane boundaries are not obvious. And that's something we'd really like to approximate in order to model the tissue and the individual cells within it in the downstream analysis. So a lot of approaches take sort of approximations based on DAPI stains, and obviously would like to capture cytoplasm as well, and that's kind of at the core of the method. If you don't do this accurately, you end up having sort of admixtures to the cells, and that's something that I guess analogous to the single cell methods where you can get sort of doublets or partial doublets, right? So basically signatures that are impure for some of the segmented cells. Okay, so before we get to segmentation, just one very simple technique that we found to be very effective and pleasing to the eye. So the idea is that we'll have the molecules here. So every point here is a molecule of a particular gene. And what we'll do for every molecule, we'll calculate a neighborhood, just nearest neighbors on the plane, and we'll calculate the profile vector, right? So what types of other molecules were seen in that particular neighborhood. And then essentially we'll embed this into a 3D color space. And when we will recolor every molecule according to that embedding in this 3D color space. And that ends up showing things like this that you see here on the bottom, where you can sort of discern, at least by eye, the different cell types and as well as their extent. So this is a very simple technique. That's why we like it. We're not cleaning it up. There are all sorts of things you could try to do to make it even better, but this gives us an assessment of the actual signal in the data. And it's pretty tough to mess it up, right? So most likely it's not a computational artifact. If you see something, it's an actual reflection of the signal in the data. So a key thing to point out here, the reason why we're seeing these cells is because the composition of the actual cells, transcriptional composition, tends to be stereotypical, and it provides a major signal. So in our method, this will be sort of a main clue. We'll try to use the transcriptional composition to provide a better guess for the extent of the cell. Here's another example. We take these randomly colored molecules, we translate it into this color space. This is a tougher tissue, so cells are smaller, closer spaced apart. For some of the cells, you actually start to see them, but there are other cells, for instance, here that are of the same type, and obviously we can't distinguish them by composition. So it's not a save-all technique, but it's helpful. Interestingly enough, so this is applying to OSMFISH, and here the columns are these neighborhoods, right? So individual molecules basically clustered, and then the clustering metadata here on the top shows the actual annotation from the OSMFISH paper from Stell Linnerson. And so the clusters more or less capture the cell types that were annotated manually in this case. And you can also project them on UMAP and essentially treat them as if they were little cells, right? Single cell RNA-seq cells, and apply, regular single cell RNA-seq methods. But as you might imagine, this is rather imprecise because these neighborhoods they'll have impurities, although obviously the area of the cell will typically be larger than the circumference, so you'll have more molecules that are sort of with pure or better neighborhoods. But you can separate these major populations, but still it's not a replacement for an actual segmentation because you're overcounting cells and the admixtures are clearly there. So the goal is to take a set of molecules and then estimate essentially the boundaries on these molecules to split them by cells, right? And I'll just again repeat the main heuristic here. A key signal that we'll use that if you have a couple of cells like this with just two genes, let's say in this case, and you have this gene in the middle, a transcript, I'm sorry, in the middle, and you'd like to figure out whether it belongs to the right cell or the left, you could actually look at the frequencies of these types of transcripts in the right and the left and decide probabilistically where it should belong. There are all sorts of parameters that you could describe to segmentation with. So some simple ones, for instance, a cell can be thought of as having a shape, which we approximate with a bivariate normal. So very simple shape, elliptical shape here. It has a composition, which is, as I mentioned, it also has a characteristic size. So all these are sort of probabilistic measures that we can combine together to evaluate the likelihood that a given configuration of molecules is actually a true cell. And there's a variety sort of different clues that we can use. So I'll just scroll through them. So for instance, as I mentioned, shape and size, we could actually get some prior clues about positions of the cell. So for instance, DAPI staining provides us with some prior information on that. We can get some prior information about the composition and cell types from, let's say, single cell RNA-seq data, and combine it all in some probabilistic way, hopefully, to optimize the cell boundaries so that they consistently optimize all these characteristics. Just in terms of technique, we rely on Markov random fields, and the optimization is done using this kind of stochastic EM algorithm with variational approximation. So here's an example just of a tissue. This is a mouse cortex measured with MERFISH, and just to show you some examples of segmentation. So different, again, the coloring is just by this neighborhood colors, right? NCVs that I showed before. But the small black lines, contours show the segmentation optimized by Baysor. And here on the right, you see the DAPI signal just for comparison. Obviously, it's important to assess whether we're doing better or not. One measure we use is just sort of transcription correlation of the disagreeing regions. So if you imagine a couple of segmentation protocols disagree in a certain region, you could take that part that disagrees between the two methods, and then just calculate its transcription composition correlation with the rest of the cells from one protocol or another, and try to figure out whether it corresponds better or not. So here's some examples. For instance, this one in the middle, OWL-FISH. So the purple segmentation is one that was originally done based on DAPI. But if you look at a compositional signal, it seems like there are actually two cells here that's separating somewhere in the middle. So these types of effects can give us some idea about how well we do, but of course, we're sort of relying on some of the same heuristic that we've built into the algorithm. Yeah, this is just more of these types of correlation benchmarks. Again, on the brain, things look very nice, well separated nuclei and signals. And this is kind of the distribution of that discrepant region correlation that we get, let's say, for MERFISH versus DAPI. DAPI in this case, just a watershed measurement. And another example from OSMFISH from Allen Institute. Again, get nice optimization of this transcriptional composition property. It can matter in terms of cell detection. So this is a quick view of OSMFISH data set, again from Stan Linnerson. And here on this embedding, after segmentation, we just generate this t-SNE embedding, and you could see in yellow are the cells that are present in the Baysor segmentation, but not in the original published segmentation. And there are some populations to which this type of approach is much more sensitive, just because it is able to pick up these types of regions where the nuclei stain, for instance, wasn't very obvious. All right. The sort of a better benchmarking, and mostly you should just focus on the top left plot here, would be again something that does not align with our heuristic. And the problem is we don't have good gold standard sort of segmentations of the cells. We don't have signal that actually defines it well. So here we're benchmarking against poly-A signal, which measures the extent of a cytoplasm, but it also can be problematic, and nearby cells can merge and so on. Nevertheless, so we use poly-A signal from this, again, OSMFISH data to approximate the boundaries, and then compare it, let's say, Watershed with Baysor. And just here, we're looking at the sort of overlap fraction for, let's say, for Baysor and then for Watershed. And you see this peak of agreement, basically, for Baysor. Nevertheless, we really wanted to be more precise. So we teamed up with Jeff Moffitt from Harvard Medical School, from Children's Hospital here nearby, to actually try to look at the membrane stains as well. And so the approach here is to measure NAKI-TPase, and it's done using an oligo-labeled antibody approach. And so here, this is IF, this membrane stain signal, essentially. This is DAPI for this region, and then this is the RNA signaling that's present there. So it does provide us with boundary signal, and it's not perfect. You still need to do computational analysis to figure it out, but it's present, and in many cells, it's actually quite good. The other thing about this data is that it's basically three-dimensional. So there's a sequence of Zstacks here, and we can start looking at how these cells show up in different Z-planes. And it becomes pretty obvious that if you're trying to segment a cell, any particular plane is going to be sparse and insufficient, and so you really need to have basically a 3D segmentation. So we've now extended Baysor to do that. The other thing that this type of data exposed is actually heterogeneity within a cell. With this resolution is rather obvious, related to both nuclear cytoplasm as well as to polarization signals within the cells. So this is just a quick overview of that tissue. It's a mouse gut epithelium. Sort of beautiful structure here, and in fact, you could go into villi and see some of the state and variation there, as well as variation within the cells. And again, we mostly use it to look at different regions with epithelial, subepithelial layers, and try to assess basically correspondence to that type of immune signal of membrane stains. This is still work in progress. We're finishing it up, but generally speaking, comparing different methods like Watershed here with this 3D Baysor segmentation or different variants of it, we're able to do reasonably well, or at least better. It's still not perfect. It seems like both combination basically of this type of combinatorial sort of composition approach, as well as auxiliary signals like membrane stain, will ultimately be needed to do the job. So here by Baysor IF prior, it's actually a combination. So we take the immunostaining segmentation as a prior for doing analysis based on transcriptional composition, and that provides the best signal, as you might have expected. All right, there are some clear challenges related to this approach. So here, a snapshot of SeqFISH, for instance, you see both very intricate cellular shapes clearly not well approximated with an oval, as well as very strong intracellular heterogeneity. Again, this is neighborhood composition signal, so it reflects the actual true composition in the cell, and you see how uneven it can be. These types of complex shapes require more advanced models that we're hoping to graduate to at some point. The other thing that I mentioned, that if you have a region with cells of the same type, then these types of methods don't really have much to go on in terms of compositional signals, so we more or less rely on the cell shape prior and auxiliary stains, such as nuclei or cell boundaries if they're present. All right, and with that, I just want to thank my collaborators. So again, Jeff Moffat, as I mentioned, that helped us with this extension of MURFISH and membrane staining. The bulk of the work, basically, the method development, is done by a graduate student in my lab, Victor Petechov, and a postdoc, Ruslan Soldatov, has helped as well. We've been helped by a lot of advice from our collaborators across the globe, including Kun Zhang from UCSD at Lane and the SpaceTX consortium, as well as Matt Nielsen that I mentioned. Thank you very much. I'm happy to answer questions first time.
Excellent. Thank you, Peter. Very interesting. So maybe I'll begin. So how do you actually use the DAPI or the immunofluorescence in the prior? Is it you're modeling the center of the cell, you're modeling the shape of the cell? How do you actually quantify this information and incorporate it in the prior here? Yeah,
that's a good question. So there are different types of prior, right? There's information about cell centers within the algorithm. So a cell has a center and it has a shape, right? And orientation, obviously, around it. So what DAPI gives us essentially is a prior on cell centers. So based on some sort of a segmentation, we can say that there's likely a cell here, but maybe we're only 80% certain of that. And the algorithm, the EM algorithm, as it iterates, it can essentially create new cells or disappear cells, and that's where DAPI becomes extremely important. Now, the IF, the membrane stain information is different because it gives us information about a potential extent of the entire cell. So we can input it as a as a segmentation, which is a prior on any two molecules within that region, the segmented region, belonging to a single cell. So that's how it comes through into the algorithm.
I see. Okay. So there is a question from an anonymous attendee, so I can't shame someone and get them to ask it in person, so I have no idea who it is. So, and the question is, how do you compare the cell and tissue? There's a mention of sequencing, so I think they're a little bit confused about how you link the SeqFISH data together with the single cell transcriptomics data, if I understand the question correctly, or how you use the two sources of information.
I see. Perhaps. Yeah, so I guess if I, at the risk of generalizing, the question is, how does single cell, regular single cell RNA-seq help us in this task? And well, the main, at this stage, when you've already measured the experiment, the main information it can give you is sort of stereotypical composition of specific cell types, right? So with single cell RNA-seq, typically you can cover a lot of cells and then derive clusters of cells and derive their transcriptional profiles. And those can be used as sort of priors for the initial clustering step to sort of separate similar cell types together and derive their shape parameters and so on. Now, it turns out that actually in most cases that's really not needed, and in some cases it decreases performance because the exact sort of proportions at which the transcripts are detected in the single cell RNA-seq can be different from the proportions that you're seeing in these targeted methods where you have only, let's say, 300 transcripts being measured, just because I think of the detection procedure and so on. So frankly, in practice, we mostly use it as a sort of side-by-side kind of validation to see if we pick up the same cell populations. But there's an option to put it in as a transcription profile prior into the method, but we basically don't use it.
Okay, so maybe whilst other people think of their questions, another one from me. So I didn't quite understand what you meant by the characteristic size, because is that the number of molecules you're talking about? Is it actually the volume? What do you mean by characteristic
yeah.
size?
So in 3D, it ends up being a volume. In 2D, I guess it would be sort of those elliptical axis radii. The cells, depending on the tissue, but you might have cells of very different size. Well, first of all, any experiment that we're given, we kind of have to figure out what the characteristic cell size is, right? I mean, if you start an algorithm outlining the potential cell size being like this, right, which is actually, hundreds or thousands of cells, it's not going to work. It's too far from an optimum, and so it's going to make mistakes. So a key feature is to sort of set the scale, right? And then when you start optimizing is to figure out how cell size varies, right? So and potentially how it varies depending on the cell type. This is an example of a tissue where I have, epithelial layer where cells are much larger, for instance, than subepithelial layer. So at the very least, we need to know a realistic range of sizes to set up the probability, well, to basically optimize probabilities correctly. It's an interesting problem that, different clues you could use. For instance, you could just take a look at the, let's say, DAPI stains, which are typically available, and just look at internuclei distance. And it turns out to be a reasonably good estimate of, of cell size distributions as well.
I see. Okay, thanks. So Tim Fessenden has a question. So cell transcript location should depend heavily on the particular transcript. Could you weight cell assignment differently for some transcripts, i.e. those that are expressed in the nucleus versus others that are expressed in the cytoskeleton or elsewhere in the cytoplasm?
Yeah, that's a great question. And it's basically a problem, right? Like we'll hopefully we'll get this out soon, this data set, and our write-up will show some more detailed examples there. But in this tissue, for instance, not only have cytoplasm versus nucleus, you also have some interesting polarization patterns. And this is kind of a biology that we wouldn't normally model in the algorithm. And yet we kind of have to account for it, because otherwise, we can segment the top of the cell, from the rest of the cell, just because in the apical part, there's some sort of special transcriptional configuration. So at the moment, and, we're not entirely happy with this, but at the moment we approach this sort of in a reasonably biased manner. As you suggest, we basically assign weights to genes that show very strong nuclear cytoplasm, very strong cytoplasm or polarization configuration. So essentially, the lower the weight of the gene, the more generic it is considered by the algorithm. So it is still considered for, let's say, density analysis and other thing, but the identity of this molecule, right, as a particular gene and its influence on the composition of the cell is basically reduced. So they basically look like same color, like gray types of transcripts. That helps a lot, right? So with this approach, you can take a look, note some of these genes and reduce their weights, and that basically straightens out the segmentations. Of course, what we're not happy about is that, if we take unknown tissue, it will take some expert, looking by eye to figure out what transcripts are showing really odd patterns like this. However, the other hope is that, as these methods scale up in terms of numbers of transcripts, different transcripts they're measuring, as we move from 100, let's say, transcript to 1,000 or more, these sort of special genes will get diluted. In this case, for instance, the panel actually included some of them on purpose, and so they sort of have an outweighed influence on this. But hopefully in the future we'll have sort of more general approaches to this.
Excellent. And maybe a final question for me. So the sort of the classical way people, well, classical, such a new field, but people would use machine learning style approaches for doing this problem. And I noticed in the comparison you have here, you don't compare to, I don't know, Elastic or one of these other machine learning approaches where you use a training set and then apply. Have you done that? Can you quantify how much of an improvement do you think Baysor yields over some of these out of the box, if you will, ML approaches?
Yeah, so what we do use and compare with are out of the box approaches on traditional stains. So for instance, IF segmentation or DAP segmentation is here done using cell pose in this case. there are undoubtedly, newer approaches being developed for, kind of compositional analysis as well. Those are, at least as far as we've seen, not quite there yet. I think this is very reasonable. I can certainly see how in the future this basically would be done using some sort of net training on some examples, or let's say preliminary segmentation by a method like this. Obviously it's error prone, but it might be enough to sort of train the networks to actually start recognizing, even particular patterns to this specific tissue better.
Excellent. Okay, well, if there are no further questions, thank you very much.